Hello, good afternoon to all of you. Good afternoon to Mrs. Tris, and good afternoon to all of our classmates that are all here this afternoon. So we are the group 2, and for today's video, we will be discussing the chapter 11, talks about the learning, memory, and amnesia. And to give you a better overview of what is the chapter all about, the reporters will discuss the following: the amnesic effects of bilateral medial temporal lobectomy, the amnesia of 𝐾𝐈𝐒𝐂𝐒𝐿 syndrome and Alzheimer's disease, amnesia after concussion, the evidence for consolidation, evolving perspective of the role of the hippocampus in memory, neurons of the medial temporal lobes and memory, where are the memories stored, synaptic mechanisms, learning and memory, and lastly, the biopsychology of memory and you. And to start the discussion, may we have Miss Catherine Joy T. Panta to take on the presentation.
Let's proceed about the lesson 11.1, which we have the objective to describe the specific memory test used to assess HM's anterograde amnesia. So first, we might know who is HM. So Henry Gustav Molaison, often referred as HM, is a famous case of anterograde and retrograde amnesia in psychology. HM suffered from epileptic seizures believed to be caused by a bicycle accident that occurred in his childhood. The seizures started out as a minor at the age of 10, but they developed a severity when HM was a teenager. So HM is quite likely the most well-known single patient in neuroscience history. His profound memory impairment caused by experimental neurosurgery to control seizures was the topic of the 5 decades of research until his death in December 2008. So in attempt to control his seizures, HM or Henry underwent brain surgery to remove his hippocampus and amygdala. As a result of this surgery, HM seizures decreased, but he could no longer form new memories or remember the prior 11 years of his life. So he lost his ability to form many types of new memories, which we called anterograde amnesia, such as new facts or faces, and the surgery also caused retrograde amnesia, as he was able to recall childhood events but lost the ability to recall experiences a few years before his surgery. So the case of HM or Henry and his lifelong participations in studies gave researchers valuable insights into how memory functions and is organized in the brain. So he is considered one of the most studied cases in medical and psychological history. So in the next slide, we can see both his left and right temporal lobes, because the removal of one medial temporal lobe has proved to be an effective treatment for patients with a lateral temporal lobe focus. So the decision was made to perform a bilateral medial temporal lobectomy, which means the removal of the medial portions of both temporal lobes, including most of hippocampus, amygdala, and adjacent cortex. So we must know what is lobectomy. Lobectomy is an operation in which a lobe or a major part of one is removed from the brain. And a lobotomy is a operation in which a lobe or major part of one is separated from the rest of the brain by the large cut but is not removed. So in several respects, HM's bilateral medial temporal lobectomy was an unqualified success. His generalized seizures were all but eliminated, and the incidence of partial seizures was reduced to 1 or 2 per day, even though the level of his anticonvulsant medication was sufficiently reduced. So furthermore, HM entered surgery a reasonable, well-adjusted individual with normal perceptual and motor abilities and normal intelligence, and he left it in nearly the same condition. Indeed, HM's IQ increased from 104 to 118 as a result of his surgery, presumably because of the decline in the incidence of his seizures, and be that as it may, HM was the last patient to receive bilateral medial temporal lobectomy because of its devastating amnestic effects. So, in the next slide, we have memory test that is used to assess HM's anterograde amnesia. So, first is digital span plus 1 test. Next is block topping memory span test. Third is mirror drawing test. And lastly is incomplete pictures test. So, in order to characterize the HM's anterograde memory problems, researchers began by measuring his performance on objective tests of various kinds of memory. So, this subsection describes 5 tests that were used to assess HM's long-term memory. The results of these first 2 tests documented HM's severe deficit in long-term memory. So, whereas the results in the last 3 indicated that HM's brain was capable of storing long-term memories, but that HM had no conscious awareness of those memories. So, this finding changed the way biopsychologists think about the brain and the memory. So, in the next slide, we must know what is a digit span subtest. So, digit span tests your ability to remember a sequence of numbers that appear on the screen, or one at a time. So, it has subtests which is digital span forward and digital span backward. So, for the digital span forward, the child repeats increasingly longer strings of numbers in the same order as presented aloud by the examiner. So, next is digital span backward. So, the child repeats increasingly longer strings of numbers in reverse order of the presented aloud by the examiner. So, for both digit forward and background, each number is read at the rate of 1 per second. Administration is discontinued when both items from the given pair are failed. So, in the next slide, we have a block topping memory span test. So, according to Birch et al. 1998, this test has been described as the single most important nonverbal task in neuropsychological research. And also, according to Smith and Scully 1994, it is extensively used in diagnostics for the assessments of visuospatial working memory, or VSWM, but it also involves spatial attention. So, we must see the block topping memory test picture in this slide. So, in the next slide, we have the mirror drawing test. So, it is a psychological assessment used to measure the rate of learning, hand-eye coordination, and neuropsychological damage. So, this is a tool to analyze and observe the nature of learning process and in studying the relationship of spatial and motor learning. So, we can see an example picture of mirror drawing test in this slide. So, in the next slide, we have the incomplete picture test. So, it is a nonverbal test or subtest often included in an IQ test in which the respondent try to identify or fill in a part that is missing from a picture of a familiar object, such as pig without a tail and revolver without a trigger. So, we can see also a example picture of incomplete picture test in this slide. So, for the next slide, we have the lesson 11.2, which we have the objective to describe 3 major scientific contributions of HM's case. So, HM's case is a story of personal tragedy, but his contributions to the study of the neural basis of memory were immense. The following 3 contributions proved to be particularly influential. So, first we have by showing that the medial temporal lobes play an especially important role in memory. So, HM's case challenged the then prevalent view that memory functions are diffusely and equivalently distributed throughout the brain. In doing so, HM case renewed efforts to relate individual brain structures to specific mnemonic or memory-related processes. So, in particular, HM's case spawned a massive research effort aimed at clarifying the mnemonic functions of the hippocampus and other medial temporal lobe structures. So, second is the discovery that bilateral medial temporal lobectomy abolished HM's ability to form certain kinds of long-term memory without disrupting his performance on tests of short-term memory or his remote memory. So, remote memory is a memory for experiences in the distant past. So, supported the theory that there are different modes of storage for short-term, long-term, and remote memory. So, HM specific problem appeared to be difficult in memory consolidation. So, the third one is the HM's case was the first to reveal that the amnestic patient might claim no recollection of a previous experience while demonstrating memory for it by improved performance. So, for example, is on the mirror drawing test and incomplete pictures test. So, this discovery led to the creation of two distinct categories of long-term memories. So, which we have conscious long-term memory became known as explicit memories or declarative memories, and long-term memories demonstrated by improved test performance without conscious awareness became known as implicit memories. So, as you will soon learn, this distinction is for general relevance. So, many people with amnesia might lose their ability to form explicit memories while maintaining their ability to form implicit memories. So, in the next slide, we have the lesson 11.3, which we have the objective to discuss what research on medial temporal lobe amnesia has taught us about learning and memory. So, in this slide, we have the question that why is the medial temporal lobe important for our memory? So, medial temporal lobes and medial diencephalic structures are thought to play a crucial role in declarative memory. They serve as a temporary deposit of information and the final storage being at neocortex level. So, in the next slide, we have the question of what research on medial temporal lobe amnesia has taught us about the learning and memory. So, research on medial temporal lobe amnesia has shown that HM's difficulty in forming explicit long-term memories while retaining the ability to form implicit long-term memories of the same experience is not unique to him. So, in the next slide, which we have the lesson 11.4, and we have the objective to describe differences between semantic and episodic memories. So, first is, we must know what is semantic memories. So, it is explicit memories for general facts or information. Next is, what is episodic memories? It is explicit memories for the particular events of one's life. So, we must know that the semantic and episodic memories are the two varieties of explicit long-term memories. So, people with medial temporal lobe amnesia have particular difficulty with episodic memories, and in other words, they have difficulty remembering specific events from their lives, even though their memory for general information is often normal. So, although they can't remember having lunch or going to a movie, chatting with a friend, or attending a lecture, they often remember what their friends are like, or a movie they have seen, a language they learn, writing, world events, and the sorts of things learned at school. So, for the lesson 11.5, which we have the objective to describe the case of RB and the phenomenon known as transient global amnesia. So, first, we must know who is RB. So, patient RB experienced an ischemic event or we known as stroke that destroyed his ability to form new memories. So he especially lost his declarative memories, and he is not able to learn new facts. He could, however, learn the ability to perform new motor tasks. So according to Sola, Morgan, Squire, and Amaral 1986, a person who have experienced an interruption of blood supply to their entire brains often suffer from medial temporal lobe amnesia, and RB is one of such individual. So, a transient ischemic attack or TIA is a stroke that lasts only few minutes. It happens when a blood supply to part of the brain is briefly blocked. Symptoms of TIA are like other symptoms of stroke but not last long, or it didn't last as long as stroke. So in the next slide, we have the question of what is transient global amnesia. So it is defined by its sudden onset in the absence of any obvious cause in otherwise normal adults. So it typically lasting only 4 to 6 hours, and this condition usually occurs in people between the ages of 50 to 70. So transient global amnesia is sudden and temporary episode of memory loss that can be attributed to a more common neurological condition such as epilepsy or stroke. So during an episode of transient global amnesia, your recall of recent events simply vanishes. So you can't remember where you are or how you got there. So how common is transient global amnesia or TGA? So TGA occurs in approximately 3 to 10 people out of every 100,000 people. So in the next slide, we have the question of what causes transient global amnesia. So we have in this slide physical exertion, emotional and psychological stress, sudden immersion in cold and hot water, head trauma, pain, sexual intercourse, and performing the Valsalva maneuver. So this is a breathing technique that purposely performed by a person as a part of medical test or to slow a rapid heart rate and other uses. It is performed by closing your mouth and pinching your nose and bearing down as if having a bowel movement. So one type of TJA results from excessive alcohol consumption, large doses of barbiturates, and illicit or street drugs, or small doses of benzodiazepines. So the next slide, we have the symptoms of transient global amnesia. So first, we have the person who have transient global amnesia usually have headaches, dizziness, nausea and vomiting, anxiety, confusion, and tingling in the feet, legs, hands, or arms. So a person with transient global amnesia involves a sudden inability to form new memories. Some people also cannot recall memories from hours, days, or longer in the past. People experiencing a TGA episode may appear disoriented and confused. They know who they are and know their friends and family members, but may repeat questions about the time or the date. Other complex mental tasks, such as the ability to drive a car or cook, are not affected. So in most cases, TGA episodes last from 1 to 10 hours. So we have 6 hours in average. So in rare cases, symptoms may persist for up to 24 hours. So memory problems that develop gradually or last for more than a day are not part of the TGA and are likely related to other causes. So for the next slide, we have here how transient global amnesia diagnosed. So we have blood test, also we have brain computed tomography or CT scan, And lastly, we have brain magnetic resonance imaging or MRI scan. So, the next slide, we have another trivia that the transient global amnesia cannot be treated or there are no treatment for the TGA. And that's all for my part of this report about the lesson 11 to 11.5. That's all and thank you.
So let's talk about amnesias of Korsakoff syndrome and Alzheimer's disease. So starting off with the first one, Korsakoff syndrome is a chronic memory disorder caused by severe deficiency of thiamine. Korsakoff syndrome is most commonly caused by alcohol misuse, but certain other conditions can also cause this syndrome. So starting with the severe thiamine deficiency, several genetic variations, poor nutrition, and sometimes be associated with disorders other than alcohol misuse, including anorexia, overly stringent dieting, fasting, starvation, or bariatric surgery. So the main symptoms would be problems learning new information, inability to remember recent events, and long-term memory gaps. So most patients first develop a delirium marked by confabulation, gait, gait, gait disorder, and problems with memory and disorientation, which is usually called Wernicke's encephalopathy. So when this delirium clears, a profound disorder of memory remains. Patients have a great difficulty learning and retaining new information, as well as problems recalling memories from the recent past. So let's talk about the Alzheimer's disease. Alzheimer's disease is a brain disorder that slowly destroys memory and thinking skills and eventually the ability to carry out simple tasks. So the exact cause of Alzheimer's disease aren't fully understood, but at the basic level, brain proteins fail to function normally, which disrupts the work of brain cells, which are the neurons, and triggers a series of toxic events. Chemically, not a relationship or something. So neurons are damaged, lose connection to each other, and eventually die. So the damage most often starts in the region of the brain that controls the memory, but the process begins years before the first symptoms. So the loss of neurons spreads in a somewhat predictable pattern to other regions of the brain. So by the late stage of the disease, the brain has shrunk significantly. So ano nga ba mga symptoms nito? So the symptoms of these are occasional memory lapses, but the memory of loss associated with the Alzheimer's disease persists and worsens. So difficulty in concentrating and thinking, especially about abstract concepts such as numbers. So symptoms may vary depending on what level of Alzheimer's the person has. So with the mild, there will be memory loss, poor judgment, leading to bad decisions. In the moderate, increased memory loss and confusion, inability to learn new things, and lastly, to the severe, inability to communicate, weight loss, and eventually would lead to seizures. So summarize the effects of a closed head injury on memory. So someone is in a coma is unconscious and will not respond to voices, other sounds, or any sorts of activities going on nearby. So the person is still alive, but the brain is functioning at its lowest stage of alertness. So common misconception is that coma would last for months or years. So the thing is, being in a coma is less about how long you sleep and more about how long you're not being able to be woken in. So you can be unconscious for a long time and still be woken up easily. It's what's called sleeping, by the way. So coma, on the other hand, would be the opposite. You could be unconscious for minutes, but cannot be woken up immediately. Okay, so let's say a closed head injury happened on a person. So this is what commonly happens. So you can refer to the picture or image in the screen. Retrograde amnesia would be experienced on the event leading up to the blow, and anterograde amnesia on the events after the blow, and there will be subsequent period of confusion. So note, more severe blows to the head tends to produce longer comas and long periods of confusion and long periods of amnesia. So let's describe the classic view of memory consolidation and some of the evidence it rests upon. So contrast the views with the current view of the memory consolidation. So classic view of memory consolidation would be Hebb's theory. So the classic theory of memory consolidation is Hebb's theory. So basically what the theory is that it explains that the memories or experiences travel in a loop on closed circuits, although these circuits are prone to disruption. Fortunately, this disruption would fortify them, which then creates a more stable long-term storage. So basically, what doesn't kill you makes you stronger, I guess. So additionally, there is what we call ECS. ECS is electroconvulsive shocks, which is an intense, brief, diffuse, seizure-inducing current that is administered to the brain, although large electrodes are attached on your scalp. So often used for more ethical experiments with regards with the memory consolidation. So ito yung gagamitin mo instead of bashing someone in the head just to create disruption. So the current view of memory consolidation would be, in other words, that the evidence indicates that elastic memories become more and more resistant to disruption throughout a person's life. Each time a memory is activated, it is updated and linked to additional memories. So these additional links increase the memories resistant to disruption by cerebral trauma, such as concussion or ECS. So in the current view of consolidation, it also introduces the idea of hippocampus playing a big role, and there is the rise of reconsolidation. So let's describe the delayed non-matching to sample test for monkeys and rats. So bear in mind na I'll just explain what is seen on the screen and kung ano nangyari sa experiment. And explain din naman mamaya nila Mary Angela and Jasmina kung ano ba naging result. A deeper and much better explanation. So in this test, a monkey is presented with a distinct object, sample object, under which it finds food. So then after a delay, a monkey is presented with two test objects, a sample object and an unfamiliar object. The monkey must remember the sample object so that it can select the unfamiliar object to obtain food concealed beneath it. So similarly, the test for rats would basically be the same as the monkeys, but would be performed on what they call a Bombay box. So on both the tests, different results were seen on the test subjects were simply trained and those who underwent bilateral medial temporal lobectomies, by the way. So those test subjects who underwent bilateral medial temporal lobectomies have the same result after the experiment, which is having an object recognition deficit.
Neuroanatomical basis of the object recognition deficits resulting from bilateral medial temporal lobectomy. So to what extent are the object recognition deficits following bilateral medial temporal lobectomy a consequence of hippocampal damage? So various effects of lesions to the medial temporal lobe structures were assessed even in the early 1990s. So performance in the delayed non-matching to the sample test in monkeys and rats are being studied. There have been challenges stated that the hippocampal damage is critical in medial temporal amnesia. Researches such as those of Bassi and Saxida in 2005, Duba and Kornikuk and Pinel in 2000, and Mambi in 2001 have reached same conclusions. Severe and permanent deficits in performance on the delayed non-matching to sample test and other tests of object recognition were due to bilateral surgical removal of the medial temporal cortex, while bilateral surgical removal of the hippocampus only resulted in modest deficits, and the bilateral destruction of the amygdala resulted to none. Figure 11.13 shows the comparison of the effects of medial temporal cortex lesions and the hippocampus plus amygdala lesions on object recognition memory in rats. Lesions on the medial temporal cortex caused severe disruptions on object recognition memory but only moderately in hippocampal lesions. This caused an increased interest of the case of RB, which was previously discussed in this chapter. He was left amnesic after the ischemic accident that occurred during heart surgery that revealed that obvious cell loss was restricted largely by CA1 hippocampal subfield. These have been further studied in monkeys by Sola Morgan et al. In 1992 and in rats by Woods et al. In 1993 that resulted in global cerebral ischemia leads to a loss of CA1 hippocampal pyramidal cells and severe deficits in performance on the delayed non-matching to sample test. The relationship between ischemia-produced hippocampal damage and object relation deficit in human monkeys and rats supported that the hippocampus plays a huge role in object recognition memory. According to Mombi et al. In 1996, damage to brain structures other than the hippocampus contributes to the amnesia seen in patients that went through global cerebral ischemia. This proved that though the damage following cerebral ischemia is in CA1 subfield of the hippocampus, there is a huge damage to other areas that is more diffuse and difficult to quantify. Allen and colleagues in 2006 found out that anterograde amnesia is most likely the result of those ischemic patients with great reduced hippocampal volume, and they also have extensive neurocortical damage. The hippocampus was initially thought to be the temporary storage of newly formed memories, but it was soon discovered that the structures of the medial temporal lobes have specific functions as they play a key role in explicit episodic memories. The adjacent medial temporal cortex is significant in the object recognition memory than the hippocampus. Neurons of the medial temporal lobes and monkey. The study of the effect of damage due to the medial temporal lobe suggested that it plays a huge role in memory, and the study of the responses of the medial temporal lobe neurons have been also enlightening. First major studies of the responses of the medial temporal lobe neurons focused on the study of the hippocampal neurons in rats. The study resulted that the bilateral regions of the hippocampus disrupt the performance of memory that's involved in spatial location. Morris-Water-Maze test, or this is where intact rats learn to swim in circular murky water placed at a various location to a stationary platform hidden just below the surface. So rats with hippocampal lesions learn this task with great difficulty. Radial arm maze test, this is where an 8 arms radiate out from a central starting chamber, and the same few arms are baited with food each day. Rats with the hippocampal lesions learn to visit those arms that contain food and do not visit the same arm more than once a day. The ability to visit a baited arm is measured by reference memory. This memory is for general principles and skills needed to perform a task. The ability to not visit an arm more than once a day is measured by working memory. So this is a temporary memory that is needed for successful performance of a task one is currently working. Rats with hippocampal lesions showed poor on both the reference memory and the working memory of the radial arm performance. Hippocampal place cells and entorhinal grid cells. Hippocampus plays a role in spatial processing because many hippocampal neurons are place cells. So place cells are neurons that respond when a subject is in specific location. So, for example, in an unfamiliar test environment, one of the hippocampal neurons of a test rat have a place field in that environment. And then as the rat familiarizes itself with the environment, those hippocampal neurons will acquire a place field in it that only fires when the rat is in a specific part of that environment. Place cells have been identified in different species, including human. So when a rat is placed in an unfamiliar test environment, it is possible to know where the rat thinks it is based from the route it takes to get in a specific location where it had been rewarded. So a study conducted by QB et al. In 2007 showed that the firing of a rat's place cells is where the rat thinks it is in the test environment, not where it actually is. So for entorhinal cortex, the major source of neural signals to the hippocampus in the area of medial temporal cortex. It was also in line with the researches on hippocampal place cells. Hippocampal place cells acquired their spatial information came from the discovery of grid cells. So these are entorhinal neurons that have an extensive array of evenly spaced place fields that produces a pattern similar of a graph paper. So the spacing between those that evenly spaced place fields can be flexible, such as of those that smaller or oddly shaped animals. So their fields are closer together or shared. Spatial computation can be done due to the even spacing of the place fields in the grid cells. These grid cells can also be seen in human patients. Other types of neurons in entorhinal cortex that is associated in spatial location are head direction. So these are tuned to the direction of head orientation. And also border cells. This is when subjects and also border cells. This is when this fires when the subject is near the borders of its environment. Evidences suggested that the responses of hippocampal place cells depend on the input from entorhinal grid cells. First, there is a pathway from entorhinal cortex to the hippocampus. Second, entorhinal grid cells respond reflexively to location, while hippocampal place cells respond to place in combination with other features of the test environment. But the discovery of hippocampal place cells emerged in rat pups prior to the appearance of stable entorhinal grid cells firing takes up to the idea that input from entorhinal grid cells is essential for hippocampal place cell functions. There are also evidences found that place cells can still function after the elimination of entorhinal grid cells. Comparative studies of the hippocampus and spatial memory. So researchers on food-catching birds have great spatial memories because they must remember the location of their food catches scattered in their territories in order to survive. According to a study by Sherry and Vaccarino in 1998, food-catching species have large hippocampuses than related non-food catching species. Though there are evidences of the role of hippocampus thus to spatial memory, the effects of its damage to spatial memory tasks in humans and other primates have been inconsistent. One problem may be is that spatial memory in humans and monkeys are tested through computer screens, whereas spatial memory of rats, mice, and birds is typically studied while they navigate through test environment. Several forms have been identified on hippocampus. So certain cells in the hippocampus are shown to time cells, code for temporal aspects of an experience. It is also involved in learning about social organization or social space in humans and mice. So in the next section, we will know that cells in the hippocampus and its surrounding structures have been shown to have a direct role in coding concepts. Jennifer Aniston neurons or concept cells. So recording electrodes are implanted in the brain of a patient with severe epilepsy, usually prior to surgery, as this record the activity of particular neurons in patients as they perform tasks. These electrodes are implanted to the medial temporal lobes as they are susceptible to epileptic discharges. So as you can see in figure 11.15, and as we've learned previously, major structures of medial temporal lobes consist of the hippocampus, amygdala, and the medial temporal cortex, while the medial temporal cortex consists of entorhinal, perirenal, and parahippocampal cortices. So the neuron fired in response to the images of the actress Jennifer Aniston, but not to 80 other images. Individuals known to the patient, such as relatives, friends, and celebrities, or to other known objects, yielded a response to other medial temporal lobe neurons. But because these neurons responded to Jennifer Aniston, they all have been termed as Jennifer Aniston neurons. So these Jennifer Aniston neurons are highly selective. They only respond to a small number of test objects or individuals. Only one could be found. They are also highly invariant. If a neuron responded to a specific object on test 1, it will only respond to that object on all subsequent tests. The most remarkable feature of Jennifer Aniston neurons is that cells. So, for example, Hale Berry neuron responded to all photos of the actress, to her printed name, and to the sound of her name. For concept cells, when cells will only respond only when the viewer perceives the concepts to which the cells are attuned to. It is also found that Jennifer Aniston neurons respond to more than one concept when there is an obvious relation between them. For example, the concept cells on the second day of testing also responded to Lisa Kudrow, Jennifer Aniston's co-star in TV series Friends. So images trigger activity in circuits of concept cells in medial temporal lobe. The discovery of Jennifer Aniston neuron is a major step forward, though it is not clear how they contribute to the storage of memories. Engram cells. So optogenetics is a procedure where neuroscientists insert an opsin gene into particular neurons. Then they use light to either hyperpolarize or depolarize those neurons. This has been used in the study of learning and memory in mice. The study of Jocelyn Kohler and Frank Land in 2015 shed light on the location of neurons that maintain an engram or engram cells. The engram cells can be identified via optogenetics through two-stage process. The first stage is called the tagging stage, While an animal engages in a learning task, the neurons that are active during the learning task are induced to express opsins. So the second stage is the manipulate stage. So the previously active neurons are either inhibited or excited by using light to influence the activity of the opsin tagged neuron. Researchers are now able to observe, suppress, or activate engram cells in different parts of the nervous system. For example, researchers optogenetically reactivate hippocampal dentate granule cells that have been previously active during the encoding of a positive experience that reversed depressive-like behavior in mice. Researchers also shown that activating an engram cells lead to the retrieval of memories that are inaccessible in transgenic mouse model of Alzheimer's disease, suggesting that the memory deficits are retrieval deficits rather than encoding deficits. In mid-20th century, where modern biopsychology began, there was a major movement to know where memories are stored. This was championed by Carl Lashley. And so where are memories stored? Lashley and others used lesion method. Destruction of a structure, if it really does hold memories, then it should eliminate all memories that were acquired prior to the lesion. Lesions of a particular structure produces either no retrograde amnesia at all or retrograde amnesia for only the experiences that occurred before the surgery. The findings led to two major conclusions. The first one is memories are stored diffusely and can survive destruction of any single structure. The next one is memory becomes resistant to disruption over time. So this chapter solely focus on four neural structures that are the keys in storage of memory. So the first one is the hippocampus. Second is the medial temporal cortex that is responsible for episodic memory. Third is the mediodorsal nucleus of the thalamus. And fourth is the basal forebrain that are inflicted for the memory deficits of Korsakoff's and Alzheimer's disease. Now we'll discuss the five brain areas inflicted in memory. So the first one is the inferotemporal cortex, or the cortex of the inferior temporal cortex. The areas of the brain that are active during the retention of an experience are also the same ones active during the original experience. This is based from various electrophysiological recording and functional brain imaging. This focus attention on the functions of the sensory and motor areas of the brain. According to Deki and Tanaka in 2006, Maya and Suzuki in 2011, and Suzuki in 2010, the inferotemporal cortex has complex visual functions, while Bassi and Saxida in 2005 argued that inferior temporal cortex, in concert with adjacent perirenal cortex, is responsible for storing memories of visual input. To support this, Naya, Yoshida, and Miyashita in 2001 studied the responses of neurons in inferotemporal cortex and perirenal cortex, while monkeys learned the relation between two items in pair of visual images. When a pair was presented, the responses were first recorded in the inferotemporal neurons and then in the perirenal neurons. But when the monkeys were tasked to recall the pair, the activity was recorded in perirenal neurons before in the inferotemporal neurons. Naya and colleagues concluded that the reverse pattern of activity reflected the retrieval of visual memory from inferotemporal cortex. The next is the amygdala. It is thought to have special role in memory for emotional significance of experiences. So rats with amygdala lesions do not respond to fear to a neutral stimulus that has been followed by electric foot shock. Becara and colleagues in 1995 reported a neuropsychological patient with bilateral damage to the amygdala that can acquire conditioned autonomic startle response to visual or auditory stimuli, but had a good memory for the training. So little evidences proved it stores memory, though it appears to be involved in strengthening emotionally significant memory stored in other structures. It is also the reason why emotion-provoking events are remembered better than neutral events. So the third one is the prefrontal cortex, or the area of frontal cortex anterior to motor cortex. Patients with damaged prefrontal cortex are not amnesic as they do not display deficits on conventional tests of memory. This proves that prefrontal cortex play different roles in memory. Two episodic memory abilities are often lost by patients with large prefrontal lesions. They often display both anterograde and retrograde deficits in memory from the order of events, even when they can remember the events themselves. They also lack in working memory. So as a result, patients with damaged prefrontal cortex have difficulty performing tasks that involves a series of responses. The prefrontal cortex consists of anatomically distinct areas that have different functions. Functional brain imaging found that complex patterns of prefrontal activity are linked with memory functions. Fundamental functions for cognitive processes, action, and task management during working tasks, and other regions participate in memory processes. So the case of a cook who couldn't is the story of a patient with prefrontal cortex damage. She is the sister of Wilder Penfield, a famous Montreal neurosurgeon. She was an excellent cook before the brain damage. Afterwards, she still remembered her favorite recipes and how to perform each cooking technique, though she was incapable to cook even a simple meal as she can carry out the steps in proper sequence. And then lastly is the cerebellum and striatum. Implicit memories of sensory-motor learning are presumed to be stored in sensory-motor circuits. Researches on neural mechanisms of memory of sensory-motor tasks have focused on two structures. So the first one is the cerebellum. It is thought to participate in the storage of memory of learned sensory-motor skills through its various neural mechanisms. Eye blink response in rabbits in Pavlovian conditioning has been investigated. So a tone or the conditional stimulus is sounded before a puff of air, which is the unconditional stimulus, is delivered to the eye. After several trials, the tone elicits an eye blink. Effects of this conditioning are stored in the form of changes in the way that cerebral neurons respond to the tone. Next is the striatum. It is thought to store memories between stimuli and responses. So this type of memory is the memory that develops over many trials. It is also referred to as habit formation. Cerebellum and striatum plays a role in certain types of memory with no obvious motor component. Norton and Mangles and Squire in 1996 found out that Parkinson's patients who had striatal damage cannot solve probabilistic discrimination problem. The problem was weather forecasting game, and the task was to predict the weather by pressing rain or shine. The patients based their answers on the stimulus card presented. Each card had a different probability of leading to sunshine in their group. In contrast, amnesic patients, those that have medial temporal lobe or medial diencephalic damage, marked great in performance but had no memory of their training.
And before we briefly discuss or have a better overview what is the synaptic mechanisms of learning and memory, let us first know what is synaptic mechanisms, learning and memory individually. So first, what is synaptic mechanisms? So for us to understand and remember the concept, let us try to know it by these key points that you are seeing now. Okay, so first, how do neurons talk to one another? Well, the action happens at the synapse. This is where the point of communication between neurons or between a neuron and a target cell, for example, target cells like in muscles and our glands. So at the synapse, the stimulation or the firing of an action potential in one neuron is possible because of our presynaptic neuron or the sending neuron that causes the transmission of a signal to another neuron or on the postsynaptic or on the receiving neuron of our central nervous system. Second, at the end of the 19th and beginning of 20th century, like the nature-nurture debate, there was also a lot of controversy whether synaptic transmission was either chemical or electrical. Some people kasi thought that signaling across the synapse involved the flow of ions directly from the neuron to another. And dahil may keyword tayo na ion, we all know that this synaptic transmission is electrical. is electrical. On the other hand naman, other people thought it depended on the release of a chemical from one neuron causing a response in the receiving neuron. And again, dahil may keyword tayo na chemical, so this is a synaptic transmission that is chemically or chemical. So, dahil dichotomous yung beliefs, different scientists, researchers, or kung ano pa man, conduct, different sets of experiments, studies, and at the end, it was revealed that synaptic transmission can either be electrical or chemical. But most of the synaptic transmission are chemical, which means that these synapses communicate using chemical messengers called the neurotransmitters. So again, recap lang. Neurotransmitters, these are the molecules used by our nervous system to transmit messages between neurons or from neurons to the target cells. And it generally happens on the synaptic cleft. So, common examples of neurotransmitters are acetylcholine, dopamine, serotonin, epinephrine, and many more neurotransmitters to mention. So again, what is meant by synaptic transmission? It is the process of synapses by which a chemical or an electrical signal that is being released and or passed from the terminal of action into another neurons that generates a signal which excites, inhibits, or modulates the cellular activity. And one important feature of synaptic mechanisms is the synaptic plasticity, or this is the ability of the brain to change and adapt to new information. So the idea that synapses could change and that this change now is dependent on how active or inactive the synapses are was first proposed, of course, by the Canadian psychologist Donald Hebb in 1949. And because of synaptic plasticity's probable contribution to memory storage, it has become one of the most intensively researched topics in all of neuroscience. Well, the major function of synaptic plasticity is that it controls how effectively two neurons communicate with each other. So the strength of the communication that happens between two synapses can be likened, let's say, to the volume of a conversation. When neurons talk, or let's say when we talk, diba, we do it so at different volumes. Some of us like to whisper to each other, while others like to shout. So, ganun daw mag-usap yung mga neurons. And the volume setting of the synapse or the synaptic strength can change both the short-term synaptic plasticity and the long-term synaptic plasticity. So, Mina, ano naman ba yung short-term at long-term synaptic plasticity yung sinasabi mo? Okay, when we say short-term synaptic plasticity, this is a form of rapid activity-dependent modulation of synaptic efficacy. Like, this is the rapid up or down of the volume control that helps determine how important that connection is to the ongoing conversation. So this connection is very critical to information processing. While on the other hand, the long-term synaptic plasticity refers to the information that has already been processed and that this information can last anywhere from minutes to hours, days, and even years. So the long-term plasticity is the dominant model for how the brain stores information. In other words, how one can create and remember new formed memories. So it is safe to assume that long-term plasticity is the main responsible in forming memories. Okay, so what is learning naman? So I have key points to remember to better understand what is learning. So learning is a change of our skills, knowledge, and behavior resulting from our different experiences. So I want you guys to remember that our brain controls everything that we do. So the more we are eager to learn something new, the stronger the neural pathway is created in our brain. And of course, at first, aminin natin that we really are struggling in learning something new. But continuous learning turns this into an effortless mastery talaga. And the process by which the brain strengthens the neurological pathway is called the hyperplasticity or the neuroplasticity. So this is the capacity of our neurons or in our neural networks in the brain to change their connections to adapt to new information or the new learnings. Okay, memory naman. On the other hand, it is the capability of our brain to hold on to the past or currently happening events and different pieces of information that can be utilized in the future at the time of the need. So how does memory work? So there are three main processes that characterize how memory works, and these processes are called encoding, storage, and retrieval. So encoding, it refers to the process through which information is learned. So how information is taken in, understood, and altered to better support storage. So we have four methods how information is usually encoded. These are the visual encoding, acoustic encoding, semantic encoding, and lastly the tactile encoding. Storage, it refers to how, where, how much, and how long encoded information is retained within our memory system. So it highlights the existence of the short-term memory and long-term memory. So encoded information is first stored in our short-term memory, but if the information is important, it can be transferred to long-term memory. And to give you a better overview of the differences between the short-term memory and long-term memory, tatandaan that the information in the short-term memory is primarily encoded acoustically, while the information in the long-term memory is primarily encoded semantically. Information in the short-term memory lasts for an average of 15 to 30 seconds, while the information in the LTM lasts for an indefinite duration of time. Third one is the information. Short-term memory has a capability lang na mag-store ng 5 to 9 items or 5 to 9 information. While the long-term memory, of course, unlimited storage of information yan. So, okay, last is the retrieval recall. So, this is the process through which individuals access stored information. So, it is said that information stored in either short-term memory or long-term memory are retrieved differently. Short-term memory is retrieved in a sequential or order manner, while the long-term memory is retrieved through association. And I guess that's it for the synaptic mechanisms, learning, and memory. Going back, this chapter mainly focused on the particular structures of human brain that is associated in learning and memory, and what happens if these specific structures of the brain are damaged. But now this part of the module focuses on the neuroplastic mechanisms that are thought to be the fundamental basis of learning and memory. And as I have discussed a while ago, most modern thinking about the neural mechanisms of the memory began when Hebb argued that there are changes in the synaptic efficiency that were the basis for long-term memory. So this part of the chapter mainly focuses exclusively on the synapse, or the point of communication between neurons. And it also deliberately talked about the importance of the long-term potentiation. So, let's discuss this long-term potentiation by giving a scenario. So, to start, paano nga ba tayo natututo? Well, for me, this is really a tougher question than it seems. We know that we learn by studying, for example, but what changes occur in our brain when we read a chapter, let's say one personality theory for the first time? When we test ourselves, kung may matatandaan ba tayong kahit ilang concepts about that personality theory that we have read. Well, we know that our brain doesn't just grow new cells to store some information in our memory system. Otherwise, our head will be, bigger and bigger. But what really happens is that connection between neurons strengthen, and this process is now called the long-term potentiation, or the LTP, and it is one example of the synaptic plasticity. Again, synaptic plasticity is the ability of the synapses to change their strength to better adapt to new information. Okay, let's try to know more about the LTP by this figure 11.8, which illustrates three hippocampal synapses at which LTP is commonly studied. And the species that has been most frequently studied is the hippocampus of a rodent. First, hippocampus is an important part of the limbic system, especially in the consolidation of the short-term memory to long-term memory. So, and also this figure is called the trisynaptic circuit of the hippocampus, and because this involves three major cell group. First, the granule cell of the dentate gyrus. Second is the pyramidal neurons of the CA3, and lastly, the pyramidal neurons of the CA1. So the circuit begins with the axons of the perforant pathway, which goes to the entorhinal cortex of cortex to the dentate gyrus, transmitting signals from the parahippocampal gyrus. Then the dentate gyrus will then transfer the signal to the pyramidal cells of the CA3 by the mossy fibers. Then finally, the CA3 will signal the cells of CA1 via the Schaffer collaterals. So again, to summarize, there are three parts of this circuit. Synapse 1 will be the enteral cortex to the dentate gyrus via the perforant pathway. Synapse 2 from the dentate gyrus to the CA3 by the mossy fibers, and synapse 3, or the last synapse, from CA3 to the CA1 via the Schaffer collaterals. Okay, let's try to dig this more by this figure 11.9. So, to be honest, medyo mahirap intindihin, but, I will try my very best to explain this in a na maintindihan ng lahat. So, this figure illustrates the LTP in the granule cell layer of the rat hippocampal dentate gyrus. So, they did a coronal section to the rodent's brain so that the spinal fluids of the section can stay alive, or the neurons can still function, ensuring that yung response will be a physiological situation. And first, what they did is they simulated the perforant pathway and recorded the response from dentate gyrus granule cells. So the perforant pathway comes from the entorhinal cortex to the hippocampus, and as I have said, the response were recorded from the granule cells when they are stimulating the perforant path. But napansin nila that each time they give a high-frequency stimulus or pulse or current using the electrodes, they have seen that there is an increased, in the postsynaptic potential slope, or the excitatory postsynaptic potential, or the EPSP slope, and they can see that the posted response lasts for several hours, and thereby they turn this kind of experiment as long-term potentiation. So, we have here again some key points to remember about long-term potentiation. First question, why LTP is the most widely studied neuroscientific phenomena? So, the reason for this is because of Hebb's highly influential theory about the memory. And if you remember, he argued and hypothesized that changes in the synaptic transmission can explain why long-term memory exists. And as for Hebb, LTP has two key properties as associated in the physiological mechanisms of both learning and memory. First, long-term potentiation can last for many weeks, even years. Second, LTP only occurs if presynaptic firing is followed immediately by postsynaptic firing. Parang cells that fire together, wire together. So, yun yung tatandaan nyo about long-term potentiation. And also for Hebb, he suggested that we learn because of the occurrence of firings in the pre- and post-synaptic neurons. So, the firing of the two neurons is really important, for learning and memory. Additionally, there are some observations relating LTP to neural mechanisms of learning and memory. There are actually 8, but among these, 4 observations are actually enough to explain the relation of the two. So first, LTP is elicited by high-frequency electrical stimulation of the presynaptic neuron that is said to mimic, the normal neural activity. Second, it was observed that the LTP effects are greatest in brain areas involved in learning and memory. Third, learning can produce LTP, or long-term potentiation-like changes. Lastly, drugs that impact learning often have parallel effects on the long-term potentiation. LTP can also be viewed as a 3-part process, like how we stored information. First is the process of LTP is the induction or learning. Second, process of LTP is the maintenance or the memory, and the last process of LTP is the expression or recalling of the stored information. And now we go to the induction of LTP. No, next slide. And the best known mechanism involves a glutamate receptor known as the NMDA receptor. So NMDA receptors do not respond maximally. So tingnan nyo na lang sa figure 11.20. Robs, lagay mo sa isang slide. Okay, NMDA receptors do not respond maximally unless a glutamate receptors binds and the neuron is already partially depolarized. Okay, when we say depolarization, it means that the neuron is more likely to fire an action potential. And when both conditions aren't met, so the calcium ions will not open in its maximal state, inhibiting the influx of calcium ions. Calcium ions influx will only occur if there is the co-occurrence of both presynaptic and postsynaptic neurons that will produce long-term potentiation. So, calcium ion influx also lead to the binding of the glutamate at an NMDA receptor that is already depolarized. And also, calcium ion influx may activate the protein kinases that is said to have an impact on synaptic transmission by altering the ion channel property, causing structural changes in the existing synapses, causing long-term potentiation. And diba, sabi ko nga, changes in the synaptic transmission is important or is associated in learning and memory. Now, yun lang naman. So, we will go to the maintenance and expression of LTP, storage and recall. So again, before we go on, let's reiterate again what is storage and what is recall or retrieval. Okay, storage, it refers to how, where, how much, and how long encoded information is retained in our memory system. And in order for our memory to go into storage, it has to pass through 3 distinct stages, like the figure you are seeing now. So, this is the Atkinson-Shiffrin model, wherein stimuli is first processed on our sensory memory, or the storage of brief sensory events such as sights, sounds, tastes, and sensory memory only lasts for a couple of seconds. Second is the short-term memory that processes incoming sensory memory. So the short-term memory, sometimes called as the working memory. So, of course, may mga information tayong dinidisregard. But marami din naman tayong information na gustong maalala. And to move this information from STM to LTM or long-term memory is called the memory consolidation. So, dito nangyayaring technique na rehearsal where one is consciously repeating the information in their mind to better remember the information that they want to store. So, last process in the model is the LTM or the long-term memory, or this is the continuous storage of information. So, like the short-term memory, long-term memory has unlimited storage capacity, and this type of memory tends to be stable and can last really a long time, mostly in many years or for years. Recall and retrieval. So, memory recall, this is remembering the information or events that were previously encoded and stored in the brain. So, retrieval, this is the third step in the processing of memory, with first being the encoding of memory and second being the storage of the memory. So, retrieval of the encoded and stored memory is very important kasi if you will not retrieve those information for future use, so there's no point, diba, in storing information on your long-term memory. Okay, and for us to get to know more about the memory retrieval, let's discuss briefly its types, forms, and the brain structures that are involved in memory retrieval. So we have 3 types. First is the free recall, or this is the type of memory retrieval in which individual attempt to remember previously studied information in any order. Second, cued recall. This is the retrieval of memory with the help of environmental cues. So such cues now are often semantic. And lastly, we have the serial recall. This is the recalling of items in order in which they were presented. So remembering your cell phone number or telephone number is a good example of serial recall. So agad-agad, dito tayo sa forms of memory retrieval. So recall, it refers to the simple process of remembering something without any cues and in the physical absence of that thing. A good example of recalling is recalling the name of a person you see or you saw. So, second is the recognition. This is the identifying information of a previously known thing after seeing that thing or experience that memory again. Like recognizing and remembering someone's name by only seeing their picture. So that is an example of recognition. So recollection naman, it refers to the rebuilding or piecing together of memory. So, like remembering the details of event using your partial memories, your clues, logic. So, this is a good example of memory recollection. And lastly is the relearning. Yung relearning of relearning of the information that has already been learned in the past but is not remembered in the present times. So, brain structures. So, studies have shown that these 6 areas of the brain are predominantly involved in recall and recognition. So, we have the prefrontal cortex that is related to the retrieval attempt. The hippocampal and parahippocampal areas of the media temporal lobe are related to the conscious recollection of information. And the anterior cingulate cortex naman is related to response selection. Fourth one is the posterior median area including the posterior cingulate is related to imagery. Fifth, we have the inferior parietal cortex that is related to space awareness. And lastly, we have the cerebellum that, especially on the left side, the cerebellum is related to the self-initiated retrieval. And although not included, but increase in globus pallidus, anterior cingulate gyrus, the thalamus and the cerebellum is seen then during memory retrieval. So, maintenance and expression of LTP. Okay, and we will start the discussion by this main question. Where do synaptic or neural mechanisms occur first? So, like the debate over the chemical or the electrical in synaptic transmission, dito naman, where do LTP or the long-term potentiation begins? Is it on the presynaptic or on the postsynaptic? And after 40 years of research and studying about the mechanisms underlying maintenance and expression of LTP, medyo vague pa rin yung results ng study. However, the quest for the neural mechanisms of the long-term potentiation, maintenance, and expression has contributed to key important discoveries. First, the LTP occurs only at specific synapses on a postsynaptic neuron, and it was stated that there must be a mechanism for keeping the events at one set of synapses lang on the postsynaptic neuron from affecting nearby synapses on the same neuron. And yung specificity daw is due to the dendritic spine. And kapag daw nag-enter yung mga calcium ions sa dendritic spine, hindi daw agad to lumalabas. So, yung mga calcium ions, they exert their efforts locally doon sa loob ng dendritic spine. Okay, second discovery is that because of protein synthesis, may structural changes na nangyayari sa long-term potentiation and that underlies a long-term effect. And the discovery that LTP can cause structural changes on the structure of neurons and neuron circuit is really interesting because, for really a long time, these structures daw are thought to be static. And yung mga kind ng structural changes daw na nakita nila in the study are, let's say, yung mga increasing in the number and size of synapses, in postsynaptic dendritic spines, and change in both presynaptic and postsynaptic membranes. And yung rate down ng changes is rapid and more frequent. Lastly, the discovery that neurons undergo structural change following the induction of LTP stimulated a search for a mechanism by which a neuron's activity could also change its structure. So this led to the discovery of numerous transcription factors. So this is where the intracellular protein that binds to the DNA influences the operation of a particular genes that were activated by neural activity. So to answer the question that I have asked a while ago, so yung LTP pala talaga or the long-term potentiation begins in the postsynaptic neuron, which signals the presynaptic neuron. So in addition to this, astrocytes, not just the neurons, has seen to be involved in the long-term potentiation. Now to the second of the last part of this chapter. Most long-term okay, ulit. Now to the second Now to the second of the last part of this chapter. Okay, most LTP researchers daw kasi has really focused on NMDA receptor-mediated LTP in the hippocampus, but nakita nila or na-discover nila that LTP is being mediated by different mechanisms elsewhere pa. And much of the long-term potentiation and the neural basis of memory is talaga, sabi nila, it is still a mystery despite many researchers' discoveries. So, one discovery na nakita nila ay may flip side pala yung LTP or yung long-term potentiation, and this is called the long-term depression. It is which the synaptic connection between neurons become weaker. So, pwede natin sabihin that this is the opposing process to long-term potentiation because in LTP, connection between neurons strengthen. So, although the function of long-term depression are not still completely understood even by now, it is thought to be important to memory formation by resetting the previous synaptic changes to allow for new memory formation via the LTP. So there are several mechanisms which why LDT occurs, but again, the best mechanism that can explain this is the NMDA and the AMPA receptors. So while the long-term potentiation typically occurs after brief but high-intensity stimulation of a postsynaptic neuron, LTD can be caused by prolonged low-intensity stimulation or stimulation that occurs after the firing of an action potential. And with the rate of modest stimulation that results in long-term depression, nakita nila that there is not enough depolarization to cause widespread removal of the magnesium blockage on the NMDA receptor. However, it was also discovered na although there is a low stimulation, NMDA receptors can accept or can allow some calcium ions into the cell, pero yun nga lang, low level of calcium ions is insufficient to activate the enzymes that facilitate long-term potentiation. And because of this, nagkakaroon ng cellular cascade that causes the removal of the AMPA receptors, and as a result, this reduces the number of glutamate receptors on the postsynaptic neuron that weakens the synapse, and that resulted to long-term depression, or the communication between neurons become weakened. So, second is the metaplasticity. So this is a major regulator of plasticity threshold and therefore has a key role in keeping synapses working in a range that permits the full expression of plasticity. Again, this is the ability of our brain to change and adapt to new information. Metaplasticity also helps to keep, the neural networks operating at an appropriate level for information processing and storage. But considerable research is still needed to clarify the mechanism that underpin metaplasticity and their contribution to behavioral learning. But, change in ability to induce subsequent synaptic plasticity, nakita nila it can induce either LPT or LTT. Pero full understanding pa of LTP and its impact on memory, yung kailangan natin para maintindihan natin ng buo, yung LDT at yung metaplasticity. Okay, now to the last part of this chapter is the biopsychology of memory and you. Okay, so hypothetically, naalala mo ba yung mga episodic experiences mo during the first few years of your life? Like, generally, yung, kunyaring edad mo, 0 to 3 years old. And, kung hindi na, you have this so-called infantile amnesia, or this is the inability of human adults to remember their episodic memory from their early childhood. So, generally speaking, lahat naman tayo ay nakakaranas ng ganito. So, payo ko lang, don't panic, it's organic. Joke lang. So, going back, To test this phenomenon, Newcomb et al. In 2000 conducted 2 experiments to answer the question. Okay. Ano ba yung question na yun? So, do normal children who fail to explicitly recall or recognize things from their early childhood display preserve implicit memories for this thing? In one study of infantile amnesia, Newcomb et al. In 1994 tested a 9 to 10 years old children by showing pictures of themselves with some of their preschool classmates. Okay, some of the participants remember almost none of them. However, they do exhibit a skin conductance response associated with seeing familiar individuals. So, skin conductance response is the sweating controlled by our sympathetic nervous system, and this is the indication of psychological or physiological arousal. And sabi, this arousal daw has been found to a strong predictor of both attention and memory. In second study of infantile amnesia by Newcomb et al. In 1995 naman, is the incomplete pictures test. So first, they recruited 3 years old, 5 years old children and adults, and ang ginawa nila, they showed a series of drawing to their participants. Okay, 3 months later, the researchers assessed the implicit memory of their participants by asking to identify the incomplete pictures. So, mga incomplete pictures na, ano ba to? Kotse ba to? Upuan ba to? Or kung ano man. Although the pictures were badly out of focus, still the participants were able to identify those incomplete pictures. Second is that implicit memories was assessed by asking the participants which of the drawings they remembered seeing before. So, as a result, 5 years old and adults showed better explicit memories than 3 years old did. But, nonetheless, all these 3 groups displayed substantial implicit memory even when they had no conscious recollection of events. So, to answer the question, both of their experiments indicated that the answer is yes. So, although these early acquired memories were forgotten, but, in reality, it really have not permanently decayed from your storage or from your memory system. We can still have a memory trace of our childhood kahit fragments na lang ang mga ito. Okay, moving on, smart drugs, these are the substances that thought to improve or enhance memory or cognition. So, ano nga ba yung common smart drugs? So, we have the ginkgo biloba extracts, the ginseng extracts, multivitamins, and such. And, ikwento ko lang, nung first year college, ang umiinom ako ng ginkgo biloba extract. And, naisip ko na, baka nga makapag-enhance to ng memory. So, to better test yung hypothesis ko na yun, nag-experiment ako. Siguro second year ko na to ginawa, second year, first sem. Tinaray kong hindi uminom ng ginkgo biloba for a week, and naalala ko, this is a midterm week. So, hindi ako nag-independent sa ginkgo biloba extract, and tinaray ko kung nakakapag-enhance nga ba to ng memory or ng cognition or hindi naman pala. So, and as a result, naging mataas pa rin naman yung score ko, without even having or taking this ginkgo biloba extract on a daily basis. So, yung hypothesis ko na yon na back up, by several researchers that has shown that no purported nootropic has memory-enhancing effect in normal, healthy people. So, wag na kayong uminom kung uminom na kayo. Pero joke lang. And, so this chapter began with the case of HM, and it will end on the case of RM who experienced post-traumatic amnesia. And when we say post-traumatic amnesia, this is a transient state of confusion, disorientation, and memory loss that occurs immediately after traumatic brain injury. And a patient with post-traumatic amnesia may have difficulties with their episodic memory. So it might include amnesia for details of their personal life and might also include anterograde amnesia. And for the case of RM, he was suffering from both retrograde and anterograde amnesia, but somehow he could remember or recall few events of his previous life. Pero hindi niya maalala if he was married or where he lived or if he has a job, At sabi ko nga, one symptoms na may PTA ang isang tao is they are having difficulty on their episodic memory or experiences. Although he suffers from post-traumatic amnesia, the irony is that RM could recall a specific memory of a person somewhere in his muddled past. And yung tao na yun, it is his fellow prisoner of the present, in the present, and iniisip niya if yung tao na ba yun at siya ay may katulad na fate or parehas ba sila na kinahantungan. So, and at the end naman, RM recovered fully from post-traumatic amnesia, and as per research, recovery of post-traumatic amnesia may vary in time and depends on the amount of injury on the brain. So, post-traumatic amnesia may be either short-term or long-term, but it's hardly ever permanent. So, continuous therapy lang will return the continuous memory, and if this return, sinabi naman na the person can usually function normally again. So, I guess that's it for the discussion of the chapter 11. So, mahaba talaga, but then, sana natulungan namin kayo or may naintindihan kayo sa pinagsasabi namin. So, again, this is Group 2, and thank you very much and good afternoon.